Good evening. On Friday, the 17th of April, at approximately 2 p.m., a dog walker found a deceased female, identity unknown, in a deciduous woodland in Edinburgh. The dog walker alerted the police, who immediately dispatched uniformed officers to investigate. Upon confirmation of remains being discovered, the police assigned the case to me, Detective Inspector Jenkins, and I was appointed the Senior Investigating Officer on this case, also known as the SIO. Tonight, we will be showing you the steps real-life CSIs take to solve crimes, from what they do when they arrive at the scene to the research and extracting evidence they do at the lab. As SIO, I'll be bringing in my series of experts to explain what they do in their job and how they collect evidence. Then, collectively, we will see if we can solve this crime. Welcome to Extracting Evidence. After a scene is secured, forensic support team is called in to assess the scene. Their role is to document the scene, secure and preserve any evidence, and identify the need for additional expertise if required. When I arrived at the scene, I noticed the remains were showing early signs of decomposition. But the post-mortem interval, or PMI, time since death, was hard to establish. A forensic pathologist was called, and they were able to ascertain that death appears to be done through strangulation with some sort of ligature. As I require a more accurate time of death to conduct this investigation, I need to call in a forensic entomologist. And for that, please welcome to the scene Dr. Catherine Brown. Kat, thank you so much for coming along. Can you just give us a quick overview of your role?
Good evening, D.I. Jenkins. Good evening, everybody. Thank you all for being here tonight. My name is Kat Brown, and I'm a forensic entomologist from the University of Portsmouth. And yes, it's taken me a long time to get here. Apologies. So my role is to work alongside the forensic pathologist in helping estimate that time since death. The benefit that we have is we work with those pathological changes to see how the insects arrive at the body and how they colonize it, and then how we can estimate that most important thing, how long that body's been there.
You said pathological changes there. Can you just explain quickly what those are?
So the pathological changes that occur to a body within about 72 hours after death tend to be rigor mortis, which is the stiffening and contraction of the muscles, alga mortis, which is the cooling of the body to environmental temperature, and liver mortis, which is the pooling of the blood on those underneath surfaces. The body also starts to decay and produce some really icky smells as well.
And so your work begins after those symptoms have gone?
Absolutely. So we've got pretty good time since death estimators from those changes. After all of those have equilibrated, the best things we have got are our little flies on the wall, our little insects, and they will give us a much more accurate time since death.
So how do you estimate that time since death?
We have to find them first, and that's what I'm going to go through with you first. So here is an example of our body. Flies like warm areas. They like orifices. I have labelled a couple here. I do not need to label the other ones. You are fully aware of natural orifices. But they also will go anywhere that is warm and humid, so around the waistband where they're quite protected. And in this case, as indicated by the pathologist, we have got a neck wound, so we will have a mass of larvae there as well. Because again, the tissue's exposed, they can get in and they can feed.
Oh, so are these locations significant? Are there differences between these different locations?
Sometimes we will get different flies colonizing different areas, but in general it will be sort of very similar. Ultimately, the ones under the body, there are typical insects. The ones around the neck, they might help us indicate whether a wound has occurred. Might be that the pathologist can't determine if the body's more decomposed, but the insects in this case will be there because of this wound, this unnatural orifice.
Right, okay. So then how do you use these insects to help determine that time of death?
We have to scrummage in the crime scene first to extract these insects. So as you can see, we've lifted the body up there, sampled them using our trusty little forceps. They're not tweezers, forceps to pick these larvae up. And then we will preserve them by boiling some water and placing them on our little larvae here. That kills them instantly. So
are those real?
These are real. They're dead. They're not going to be crawling around the auditorium. They are real but dead. Best thing if you get maggots in your bins at home, boiling water, dead instantly. Fantastic. So once we've killed them, dead, gone, finished, we then put them into ethanol. And if I show you our little ethanol vial here, try and get the focal length right. So it's just a simple alcohol-based solution. Do not drink, not good for your livers. And we can then take those back to the lab. But what we also have to do is collect samples as well that we rear to adults. This is one of our rearing chambers. And the reason we rear them to adults is because maggots all look the same. All the different species of fly that we have, generally very, very minor differences between them. But the adults, we can tell a little bit more about what species they are. So we rear them in our special insectaries back in the laboratory. And
so that, can you go into a little more detail about the importance of that rearing rather than just looking at the maggots when you find them?
So we need to rear them because we need to work out what species they are. Now, every different insect that visits a cadaver is a different species. Every single one will have a different life cycle. And it's the exploitation of that life cycle biology that allows us to estimate time since death. So just an example here. This is your adult blowfly. These will be the ones buzzing around the body. They will, within about 4 hours of death, they will lay eggs on the body if the body is freely available. Eggs look like Parmesan cheese. You'll never eat Parmesan cheese the same way again. These then hatch into first stage maggots, and we've got 3 stages here. So we've got first stage, or instar as we call them, second instar, and third instar. These are your fishing maggots. These are the ones that are this sort of size. So they get up to about 2 centimetres. And they do this by feeding voraciously on the body. After about a week or so, depending on temperature, they enter the metamorphosis or pupal stage. And this is where they go from maggot into fly. And these tend to be in our lovely knocked-over plants, underneath the body, etc., where they're nice and protected. Then they will emerge again. So knowing this life cycle, being able to identify what species we've got, means we know our timings. Using the larvae that we've got on the body, we will then measure their length. And as you'll see from this graph here, the longer they are, the older they are. And this little shrinky bit here is when they go away from the body and they contract down a little bit before they enter the metamorphosis stage.
Amazing. And why did you choose these insects and not just other ones? And how can you make sure that these insects are part of the body and haven't just been there otherwise?
The disgusting thing about these insects is they have to feed on decaying matter or decaying protein, that sort of thing, to be able to develop. They need it to develop their ovaries and things like that. So they are reliant on a protein source to develop, which is why they're so attracted to bodies, because it's their life source.
And so from looking at these and the length of the bodies, you can give me a time since death of approximately 10 days?
Yeah, we're looking at about that. I mean, you're asking me to guess just from eyeballing, but yes, I would say based on what we've got, and Calliphora vomitoria, fab name, common Scottish species, so I would say roughly about 10 days, a minimum of 10 days that body has been there.
And so what extra details would you need to get a more accurate time?
Very good question. We need lots. So, as I said, the fly life cycle is not only dependent on species, but it's dependent on temperature. The hotter it is, the faster they grow. So, in addition to temperature, we need to know the humidity of the scene. We need to know wind speeds, because they don't fly if it's too windy. We need to know rainfall. They don't like the wet, a bit like me. We need to know if it's been sunny. If it's sunny and warm, they're more likely to visit a body. So, we need all of these environmental factors first before we can work out their age. I'll take the samples back to the laboratory. As I said, I'll rear the maggots up to adults. The adults will then be identified, and then I can really refine my estimate based on that data, based on that we know what species we've got, and it will probably be a minimum of about 10 days.
10 days, fantastic. And based on the evidence that you found at the scene, is there anything else that you would recommend to investigate at this time?
So when I was collecting the larvae from the neck wound, I did see that there was a bit of botanical evidence of some kind, not a botanist, in her hair. So not my remit. I would definitely recommend getting in a forensic botanist to see what's going on with the debris that looked a little bit out of place.
Brilliant. Can everyone give Kat a really big round of applause? Thank you so much for coming. Thank you. From Kat's Evidence and assistance, I now know that the death took place roughly 10 days ago. And mercifully, there was a witness statement from about that time that said they saw a disturbance roughly in the area that the body was found. They said they saw a car arrive and 2 men step out. They were able to remember the license plates as well. They say they arrived from an unknown location, but it was in a southerly direction. This might imply some sort of second scene. And to learn a little bit more about that, can we please welcome forensic soil scientist, Professor Lorna Dawson. Lorna, thank you so much for joining me. Can you just give everyone a quick overview of your role?
Thank you very much for inviting me on your scene, D.I.
You're more than welcome.
Good evening. I'm Lorna Dawson, and I'm a forensic soil scientist at the James Hutton Institute.
Brilliant. And so what do you do when you arrive at a scene like this?
Arriving at a scene, the first thing that we would do, we would look to see if there's any indication of the likely way in or out, and where someone may have made contact with the ground, or any damage to vegetation. So I would then prioritize. Here we can see the forensic markers in place, where indeed then the samples would be collected at these points and secured in an evidence bag, so that these would be seized by the police so that they could later be analysed to look for any potential evidence to link or to test for any potential link between this scene and any suspect that might be apprehended.
And what have you found that's interesting in this situation?
Well, I was speaking to the forensic entomologist, and she said that she'd noticed that there was vegetation and material within the hair of the victim, and also within the fingernails, that was not natural to this environment where the victim was found.
So you've got here a bunch of different markers of areas where you're going to look, but even though I'm a fantastic DI, they just look like a standard woodland to me, and that's a tree. So how do you know what to look for, and how do you know what bits to take your investigation to?
So the team is full of experts of geology, botany, soil science, and what we do is we look at that scene and see if there's any changes to what would naturally be there. And this is, trust me, it's a deciduous woodland, so that there's a specific type of soil that would be there. It's based on its geology, its place in the landscape. And what's been done here is soil samples have been taken, as I demonstrated here, to represent that area. So we've got this crime scene covered in terms of representing its heterogeneity. So the samples are taken, secured with a sterile spatula, and put in an evidence bag. And again, we'll look at the footwear, and in fact, here we can just look a little bit closer. And if you look at the soles of the victim, I think you'll agree that that doesn't look like deciduous leaf material, does it?
What bit are we looking for there, Lorna?
So you see the green needle?
Yes.
That is a conifer needle, and it's likely to be Sitka spruce.
So you've managed to collect some interesting materials here. How does that potentially involve a second site? How would you narrow down where on earth these materials have come from?
So we would look at this material, the soil and the vegetation. We would describe it, and we would then also analyze it. Analyze it with a suite of different analytical techniques in our accredited laboratories, and using the appropriate methods, the data would relate to the vast database that we've got that covers Scotland. And what we can do is we can use that data, link it to the data that's held spatially, the geospatial data, and we can put that sample result for that unknown sample, that sample that's on the victim's boots that does not match the area that she's found in. This could potentially be another site. that is interesting for you to investigate.
And how accurate can you get these site locations? What's the sort of scale we're looking at here?
Well, we can combine a whole range of attributes: soil elemental data, mineralogy, organic characteristics, pollen profiles. We can add all them up, and that gives us so much information. And all that data is also on this database, this GIS model that we can run.
So can you explain what GIS is?
A GIS is a geographical information system, and it's basically a system that is spatial, and we can put those layers of data. And what I've done here for you, D.I. Jenkins, is I've run a model which is based on the site where the victim was found. And what we've done here is we've run it within 30 minutes traveling time along the road south, where the victim, I believe, told you that the car had been seen coming from the south
yes.
to this site. So it all starts to fit in. All the evidence is coming together. So we look at that area. We add in all the soils that have a pH. That's how acid or alkaline it is. And this is acid, 3.85, I think, the soil on the boot, and 3.87, the soil on the fingernails. So we can put the layer of pH, like the old acetates on your systems that you used when you were projecting. So pile on that information till we narrow down and narrow down. Here we know it's a peaty soil, so we can click in all the soils on top of that pH, on top of the travelling time. And here's the distance to road, and we know the layer coniferous woodland. And what do we have left? We've about 5 areas in the whole of that area south of Edinburgh that matches the characteristics that were on that boot.
Wow. And so we can narrow our search down to very, very specific geographical areas. And this is because of that database you mentioned of the soil samples across Edinburgh. How detailed is that database? What are we talking? We're talking it's miles per section, or is it kilometres, or is it metres per section of information?
Well, it depends on many things. One thing being how much data you've got, so that you can get higher resolution, and also where it is. But we've got data 1 to 25,000 scale, so we can actually go to an area such as this. So these are the 2 forests that were identified, that were Sitka spruce, that had that pH, that had the peaty soils. And these were the areas that compared in the database, and there was, you could go 500 metres, and then you were into another soil.
Oh, wow, so less than a kilometre, and you can get different soil types, and we know the differences between them.
Absolutely.
Amazing. So, with that information in these second sites, I would dispatch my uniformed officers to go there, and when they arrived, they actually managed to find some suspicious items at this second location in the woodlands that you suggested, which looked like, we found some disturbed soil, discarded underwear, and a rope. When you would arrive at this scene, what would you then do?
I would do the equivalent to characterize this site by taking these reference samples so that it would represent, so we could look for any link between anyone that's been to there, any tool, any object that's come into contact with it, to see if then we could look at footwear, vehicles, tools, clothing, and the material on that compare with either site, and indeed both.
Fantastic. Can we give Lorna a massive round of applause?
Thank you very much.
Thank you, Lorna. So having taken that information about the soil samples and be able to find a second location, now having potentially very suspicious items in there, it's incredibly important that we collect that evidence such that we can analyse it later. And to do that, can you please welcome forensic biologist Dr. Nick Dorney.
Hello.
Nick, thank you so much for joining. Do you want to give everyone just a quick overview as to what you do in your role?
Yeah, hi everybody. My name's Nick Dorney. I'm a forensic geneticist. I work at Liverpool John Moores University. I primarily help the police and advise the police on better ways to kind of secure and collect evidence. I also develop new DNA profiling methods, and I teach on the forensic CSI program as well. So I'm here in my role as a CSI today.
Thank you so much. So what evidence are you trying to collect?
Absolutely. So you've got these obvious things. You've got your discarded underwear, you've got your circular cord that has some hair in it, and you've got this area of disturbed soil. Lorna previously was talking to you about the importance of this disturbed soil in terms of being able to map that to previous locations, the boots, the material in the victim's hair as well. But then we've got questions regarding this discarded underwear and the circular cord. Are these associated with the crime? Are these just randomly discarded? We need to try and establish a link between these two crime scenes.
Fantastic. So can you find DNA on these items?
Absolutely. DNA is everywhere. We've heard from non-human specialists as well. So we've had Lorna talking about the DNA that you can recover from soil and the kind of characterization you can do of soil types. We can get DNA from the entomological aspects as well. But what we'd be looking to get here would be human DNA to establish a link between these items and the suspect or the victim.
So what do you need to do to make sure that you can collect these items in such a way that you can get the DNA off them later?
So that's primarily the role of the CSI team. It's not just one person that attends a scene. We all have unique CSI roles. There would be a CSI who would be the crime scene manager, for example, and that person's role would be to manage their team and access the crime scene and overall assess how to triage the evidence items. You'll have CSIs whose specialisms are photography, and their primary role is to just photograph and document the crime scene. And then you'll have other CSIs whose primary role is to package the items. And this becomes quite an important aspect of DNA preservation. We've got lots of different tools and lots of different equipment, but we have to select the right one for the job. And this is where we'd go back to our CSI van, we'd collect the equipment that we need, we'd carry it to the scene, and we'd start bagging the items appropriately. So what I've got here is just a selection of the kind of specialty equipment that we've got here. What we've got here is called a weapons tube. If we find things like knives on site, they go in these tubes, and they can be adjusted according to the length of the weapon, and therefore you're not going to accidentally jab yourself on the weapon during its transportation. You can
Bad.
Yes, bad. Yes, sharpy, pointy things, bad. You've got biohazards associated with these things, and so all of a sudden, if you accidentally jab yourself with, the knife, that kind of creates a lot of health and safety. We've also got other kind of packaging as well. This is actually for tin cans. So one of the things you'd want to, if you found a tin can on site, there's not one here, but if you found a tin can on site, you might want to preserve it for fingerprinting. So this tin can would sit in here, and it would basically protect that tin can. We've got a wide variety of other evidence items. This, for example, is taking for a footwear cast. So you'd be able to load your footwear cast into there. So if we saw interesting footprints at the crime scene, we'd actually take a cast there and then store it in here. And then we've got these other two. We've got paper bags and we've got plastic bags as well. Paper bags are really useful when evidence items are wet because the paper bag just allows them to dry out. We wouldn't necessarily package wet items in a plastic bag. As soon as you've got moisture and humidity associated with things, which will be increased in the plastic bag, you start getting DNA degradation, and that's what we're wanting to prevent. So looking at these items, the first thing that I would do is I'd assess the ambient conditions. How wet is it? How wet are the items that we've got here? I'd be packaging these items in either plastic bags because it's dry, or I'd be putting them in the underwear. I might put in a paper bag. The circular cord I'd put in a plastic bag.
Brilliant. Thank you. Next, so when you've now collected those items, you've taken them back, what are you looking for? Let's start with the underwear. What are you looking for when you're testing the underwear?
So again, we're trying to establish a link between the crime scenes and the victims and the suspects. So what we'd be looking to do with the underwear is actually collect some wearer DNA from the underwear. We can do this using a process called mini-taping, which George will tell you more about in the laboratory section. But we can recover wearer DNA from this material, and that will establish a link between this underwear and the victim. We'd also there might be a suspicion of some form of sexual assault, so we might look to assess whether there's any seminal material or saliva associated with the underwear as well.
And so you're saying that you can detect that sort of saliva and seminal material So how would you go about, let's say, for example, detecting saliva on a piece of underwear?
Absolutely. So saliva is detected using a presumptive test. Presumptive tests give us an indication of the presence of different biological stains on evidence. We have to be a bit careful with presumptive tests because they do also they can cross-react sometimes.
And what do you mean by presumptive test? What does that mean?
A presumptive test is something that's not diagnostic. It's something that will give us an inkling that it's something like saliva, but it won't be strong enough to say definitively it is saliva. But we do have to be a bit careful with this. There's an interesting research article that was published a few years ago where some scientists looked at the length of time you could recover saliva from different types of underwear. And in order, being good scientists, they had to have control underwear that wasn't spiked with saliva. And so they went to the shop and bought this underwear and didn't spike it and tested it using their presumptive tests. And they found saliva on the control unspiked underwear.
So
which
they found
says something.
Saliva from a shop's underwear.
They
I won't
did.
specify which shop. How did the saliva get to underwear in a shop?
Well, this became the whole rest of the discussion of this scientific research paper. How did this saliva get there? In the end, it was assumed that it was probably an accidental transfer event from someone maybe like the shopkeeper or the store. When they are hanging the clothes, it might be a achoo, and then hang, and all of a sudden you've got the same reactive materials that would cause a presumptive test to go positive.
So there's one takeaway from tonight, it's wash your clothes after you buy them from a shop.
Or don't be picky,
Yeah.
whichever.
Everyone comes first. Oh, human beings are disgusting. So we're talking about the underwear. With the rope, I'm sitting here with your evidence bag, you're actually collecting everything, not just taking a swab, not just taking the hair. Why is that? It should be quicker just to get the little bits you want.
In terms of swabbing it?
Yeah.
So we tend to swab items that are large and immovable. When you've got something like this, simply bagging the whole item gives the forensic labs the most choice in terms of how they want to process it. The thing with swabs is that they're really easy to use and they're quite popular, but they're not very effective at actually lifting DNA from surfaces. So if I swabbed something like this and sent the swab in, I'd be leaving DNA on that, which might be potentially relevant. And so it's better to bag the whole item and send it to the labs. They might choose to snip that and cut it and then extract from a part of the cord, and they wouldn't even bother swabbing it at all. That way, there's got a greater opportunity to recover more DNA from that cord.
How much DNA are we looking to recover here with these swabs?
Again, DNA is present in the myriad of different biological stains. If it's blood, you're going to have a high chance. With this here, you're talking about what's called trace DNA or touch DNA. This is some of the most complicated DNA sample types that we have to work with. The success rates of working with these is just generally really, really low, but it is possible, and we can get DNA from these sometimes.
Brilliant. And we also noticed that there's clearly hair in this rope as well. What's important to look for when you're collecting hair samples?
Yeah, so hair's really useful. We can process hair using two different DNA technologies and two different DNA methods. One of them is called nuclear STR typing. The other is called mitochondrial DNA typing. We'd want to try and approach this with nuclear DNA typing. It's a more powerful method, but in order to do that, we need to have cells and root tips on the hairs. So we'd be looking to assess the presence of hair follicles at the end of these hairs in order to select those from the number of hairs there and preferentially type those.
Brilliant, thank you so much. And how long will it take, once we've sent this DNA sampling off, to get the results back?
Again, it varies. We'll talk about turnaround times and the pressures that forensic scientists are under to kind of meet those policing demands. Best thing to do in that situation is to talk to a forensic analyst.
Brilliant. We give Nick a massive round of applause. Thank you, Nick. Oh, head dancing. So with those items collected from the second site, they're now going to be sent off to the lab to be analysed, just like Nick mentioned. And for that, we're going to need a forensic DNA analyst. So can you please welcome to the stage Dr. George Zoglianis.
Hello.
George, thank you so much for coming. Do you want to just give everyone a quick overview as to what you do in your role?
Yes, I'm George Zouganellis. I work at the University of Derby. I'm a senior lecturer there, and I do forensic DNA analysis.
So can you just walk us through how all of this works?
Okay, so basically the forensic DNA analysis has particular steps. We have talked so far about the first step where you actually collect the evidence. What you do next, you go to the lab and you try to actually extract DNA. DNA is like just any other chemical. And
how do you extract the DNA there, George? Because
you have a few years. It's a very, very difficult and specialized process. And it really depends on what the specimen is.
Mm-hmm.
Yeah.
But so you actually like physically getting DNA out of the cells itself?
Out of the cells, yes.
Right,
This is the whole point, yes.
Mm-hmm.
And you have to have it in high purity, so you can actually do a process known as polymerase chain reaction, which is an amazing technique that does two things. Multiplies DNA, but at the same time, it can specify particular pieces of DNA you want to work with downstream. So it's a multiplication process, but also selective process.
And why, George, is it a selective process? Why would we want to sequence the whole genome?
Right. So think that we are about 3 billion bases worth of DNA. We have huge similarity between us, and only about 1% we're different. So we have to find ways to actually just address these pieces that make us unique as individuals. Okay. So this technique would allow us to do that. However, even after that technique, you cannot see anything. You will bring the tube out, it would be colorless. So you have to actually somehow make a profile, and you use a technique known as capillary electrophoresis to actually, what this does is basically these particular pieces of DNA, it translates them to fluorescent signals. So what you actually print out is, what you will actually see is just a set of peaks, like any other chemical analysis. And so you have a shape in front of you, and you have what we call then a DNA fingerprint, which you can compare from a person to a person, yeah?
Perfect.
Yeah.
Thank you.
And then the most important part is what we call statistical analysis. Because it's not enough to actually say that you have a fingerprint, you actually have to prove mathematical uniqueness. So forensic genetics is like an art form between what you do in the lab, and also you need a little bit of math to get stuff done. Okay.
Brilliant. So with all of that in mind, let's start with the underwear. So when you brought the underwear back, what do your tests show?
Right. So there we go. So first thing we actually want to see is to who's the wearer of the underwear. And how do you do that? You probably go somewhere in the piece of clothing where you have good contact between the skin, yeah, and the piece of clothing. So you can see over there, you use tape, and you can pick up what we call epithelial cells, which is basically skin cells that will actually rub off the cloth, yeah? And from those, believe it or not, you have enough DNA to actually get a DNA profile. The other thing is we observe that over here, that there is a stain. So because of the nature of this type of clothing, the first thing you actually suspect is that this stain could be semen. So before you actually do an expensive technique and because to do DNA, these things cost a little bit, you need something else, which we call it presumptive test. And for that, we use something clever that us middle-aged men are very, very familiar with, which is the PSA test. You see the same substance that we actually monitor for prostate should be present in any type of semen stain. So we actually did testing there, and that was negative. So that stain wasn't semen. And as you can actually see in the next slide, we could only compare the DNA from the tape sample and the victim DNA. And we proved that, yeah, this is the underwear that the victim was actually wearing. but nothing further we could actually say about this piece of clothing.
So that piece of evidence doesn't really help us further the investigation. But what about the rope that we sent to you?
Right, so now, what is quite interesting is that there is a bunch of hair there, right? So what you can actually do, you can use classical microscopy, light microscopy, and observe the hair under the microscope, and you're looking to find hair that have hair root cells, like the one in the picture. And if you have a few cells attached to hair, that would be good enough to downstream get a DNA profile. So we did that. Also what we did, we chopped a couple pieces from the rope, and we used a method called centrifugation, and we could actually isolate skin cells. And we split these skin cells into two portions. And in the next slide, when we examined the skin cells, we found out that there is a mixture. So there is more than one people participating, donating skin cells in the ligature, whereas the hair definitely belongs to the victim. Now,
with that mixture, does that mean that we can definitely identify one person or not? Or is it impossible to get any information out of a mixture of DNA?
Now, the way things are, and with AI, you can actually combine data from the mixture and possible data you have from DNA databases, yeah? And you can possibly match a component of the mixture. However, the big problem is that you have to remember that this data has to be brought into court. And something like that is very, very difficult to communicate to a jury. What happens is that the people that you actually saw so far, and Dr. Chen, who's sitting over there, we belong in a group called Scandi, and we are developing methods where you can examine these cells one by one. Okay? As we speak, this happened. We are a research group. So what we have actually done for this case, we have kept the other half of the portion of these cells, and we examine them one by one to actually resolve this mixture.
Amazing. So we managed to get one set of DNA, which we can see matches some suspect in a database.
Yeah, I call him suspect D for the purposes of this slide.
And then there's another suspect that we couldn't match to anyone. Why is that? Why can't we match them to somebody?
Well, not all citizens have actually given DNA in the database, and there's an entire discussion about whether we should or what have you, but there's about 6 million people that are donors in this database. And I wouldn't like to actually get into the politics of it right now. Maybe later on you can ask questions, yes.
I'll put it up the chain of command, don't you worry. Right, so what we're learning here, and then from your work of separating this mixture, is that there may have been more than one assailant at the time using that rope. And as a result, it's probably important that we have a forensic strategy meeting to discuss everything we've learned so far. So first of all, can we have a big round of applause for George?
Thank you.
Thank you so much. And then take a seat. And can we please welcome back on all of our experts? So this is a forensic strategy meeting. Could one of you just give us a quick overview of what these are and the purpose of them?
So, D.I. Jenkins, it's very kind of you to ask us along to this. And the main purpose is for you to gather all the evidence and the data from the people who've been involved, the experts, the scientists that come along and gathered evidence. And it allows you to build a better picture and be informed with basic evidence.
So let's see if we can get this all together. So Kat, you helped us find the time of death, which was about 10 days or so, and that allowed us to give us that window, which we matched with a witness statement, the We talked about the car, the assailants, and driving from a southerly location. Lorna, with your soil evidence, you helped us find that second location, which had a bunch of suspicious items. Nick, you and your work allowed us to collect those items to begin that DNA research. And through your DNA analysis, we managed to get a DNA match with our our victim, who we can then, from the database, identify was a Liz Buchanan. And then we also had a DNA match with a suspect, and we actually found out when we took them into custody from the car that it was 45-year-old Alex Duncan's skin was found on the rope. So now that I've managed to get a suspect and take them into custody, what other forensic details could I now get off them once they're in with the police?
Yeah, there's you start grabbing as many things as you can, to be honest. A lot of it these days is digital evidence. We were mainly focusing on the kind of the biological aspects in the scenario we were role-playing this evening, but digital evidence is now becoming so common in crime scenes, and the amount that you can do with it is phenomenal. You can use it to triangulate, and geolocate where people were at certain time events. Lots of people self-incriminate by actually taking photographs as well, on their mobile phones. So there'll be a deep trawl of their digital data as part of that. Depending on who else, if it's a suspect you've already got a DNA profile from, you might not need to take a reference sample. But if they've got associates that you identify, you'd take a reference sample from them as well, and then you'd follow that back to see whether those profiles were present in any of the crime scene data as well.
You'd want to seize their shoes, of course, and their car, because those are vital clues. They have to get to the scene somehow.
Yeah. And in doing so, in looking through all of that evidence, we managed to correctly corroborate the fact that soil samples that you found at the second scene were also present in the car, as well as samples from the first scene. We managed to collect the phone records from Alex Duncan, which placed them, using triangulation, at the scene of the crime, where they made a phone call to a friend, who we then took for questioning, and their DNA matched the second set that you managed to collect, Willie McTaggart. So when we took them both in for further questioning, they both just kept saying no comment throughout all of that. Is that actually something that we would see normally in real life, or is that just kind of a TV thing?
Yeah, that happens all the time, especially when solicitors get involved.
Frustrating. But tonight we've had a pretty neat story there. We had all the evidence that we needed to find them. We used all of that, all cross-referenced. It was great, and it tied our story up in a neat little bow. We managed to find the people that did it. It's Alex Duncan and this William McTaggart who managed to murder Liz Buchanan. But is it always this straightforward in real life? Can we get things to be this cut and dry?
What we just described was probably the best case scenario. It's not what I just told you about, extracting epithelial cells from a rope. Probably I could do it out of 40 times, maybe once or twice. Wow. Yeah, it has like a 5 to 10% chance of actually getting there. And you have to have a lot of experience in this particular types of.
You build up some knowledge about the associated success rates at which different evidence items generally yield results. And as we said, the kind of touch and trace DNA, the success rates are so poor, but some of the other evidence as well, I mean, it doesn't work 100% of the time. You're really...
Like the example with the stain, one might hypothesize that there was something there, but there is also time and degradation and other factors.
You've kind of got a golden 24-hour window where if you manage to collect evidence within that time and stabilize it by collecting and freezing, then you drastically increase your chances of actually being able to do anything. So early crime scene attendance is really, really key. Yeah. The longer it goes, the more decay, the more decomposition, the harder it is to get any kind of biological evidence.
But you keep all your options open and you eventually exclude and eliminate. And of course, it's important to always consider alternative propositions. So we wouldn't just assume that because we had a match, that it would definitely have come from there. We've got to consider, well, where else could it have come from? And that's part of our legal system.
Well, absolutely. The whole role of a forensic scientist at the end of the day is to refute or support the evidence for both the prosecution and the defense. So there's a kind of misconception that we're just out there to kind of, do what the police say and kind of catch the bad guy, but we have to kind of be open to all scenarios in our interpretation of that data.
Yeah.
I mean, based on what we found out later on in the case, in something like this, where I came in very early on, my estimate is based on the knowledge of that being a single scene. So I was able to say, right, the insects are potentially indicating a time since death of 10 days. However, that is only subjected to my knowledge. Having known that there was a second scene, I would need to go to that scene and see if there are any older insects, because then that would change, potentially, my time since death estimate. It might go from 10 days to pupal stage 2 weeks, I don't know, 24. So it may not actually have massive pending on the case, but in terms of what I'm able to provide and accuracy and reliability, I need to know everything that's gone on to be able to say, this is my estimate.
So there are lots of forensic things that actually need to work in tandem together to be able to paint a proper picture. It's not just isolated little pockets.
Yeah.
So finally, before we go over to our fantastic audience to ask you a couple of questions as well, you mentioned your Scandi project earlier, and it's what you all collectively work on. So what sort of work are you doing, and how is that going to change sort of forensics? What is the research there?
Well, realistically speaking, most of the samples you actually collect as far as DNA is concerned in the scene of crime are mixtures. Whether they are mixtures during the crime or, because we forget to actually mention that, there is a case of contamination, right? So because of that reason, if we have a technical way of actually separating mixed profiles physically, then that resolution would be extremely helpful to say who actually contributed.
The project itself, the Scandi project, stands for a single cell analysis for DNA identification. So as Georgia points out, it's all about trying to see what we can do with a single cell. At this moment in time, when you're collecting such a biological material, it might be hundreds, maybe thousands of cells. They might be mixed, they might not be. But we still need hundreds of cells to generate DNA, meaningful DNA evidence. But technology is moving on so fast that we're soon going to be able to tell a DNA profile from a single caught cell. And if we can do that by separating mixed sample types using the technology we're developing as part of the Scandi project, it will allow us to say with a lot more confidence, this DNA, this sample was recovered from that item, and it links to that individual.
And one of the most fantastic things as well is before we even applied for the grant together, we involved the stakeholders, the forensic science service companies, the National Crime Agency, industry who make the kits, and they're working with us in collaboration and cooperation. So all through this project, we've regular meetings with the stakeholders so that hopefully, and it's really exciting, and I think we will, this could be a breakthrough in forensic science.
Yes, it's a cutting edge of what is happening in forensic science as we speak.
Yeah, because it's 45%
Not because
of
we are doing
samples
it,
that are
but-
mixtures, something like that.
And it's not just about mixed samples as well. I mean, that's our focus at this point in time, is to being able to separate all these cells. It will enable us, hopefully, to look at those more degraded samples where we haven't got enough DNA now to be able to get a profile. But if we can go back to those degraded samples, the difficult samples, I can get DNA from a maggot gut. Okay, it's not easy, but if we have got the ability to be able to separate these single cells, we've got much more chance of being able to get a decent profile from really obscure, difficult, degraded evidence. So the potential for this kind of work goes beyond mixtures.
Well, absolutely. What they're starting to do now is assess whether you can do what's called airborne DNA testing. So can you have a vacuum where you could put in a room like this, suck the DNA out of it, and then find out who was in the room? This is where the technology is going to be going in the next 10 to 15 years.
So we're on day 15 of the 14, 15 of the Science Festival.
No, but
Thousands of people have come.
Absolutely.
If we had that technology
if
during
there are-
COVID,
if
you have to remember that these applications in forensic science do not only correspond to forensic science. Like, we do something for forensic science, but many other activities are actually influenced. I mean, including unbelievable things such as archaeology or...
Well, airborne DNA testing is going to come in with hostage crises as well. So that's interesting.
There's so much we could discuss. Right, I literally could sit and chat to you all day, but it's really important I ask these fantastic people here some questions. But before I do, please give me a massive round of applause for our experts, George, Matt, Laura, and Nick. That was superb. Thank you. Thank you so much for being so ready. Great. pass over to you, lovely lot, for some questions. We've got this. It's our throwable microphone. It's great. We'll try and get through as many people
Are you good at
as
catch?
we can. I'm terrible at throwing. I just played a bad DI. Do you think I'm good at sport? Right. Anyone got any questions for our fantastic
From
panel?
here, DI Jenkins.
Here.
Right here. Those people.
Thank you very much. Doctor, I'm amazed about these DNA profiling and stuff. However, this was quite simple thing. How many features can you extract from a DNA? Like, for example, what the race of the person had been and stuff. In total, now, currently, as we stand, you know, how many features of a DNA we can see from a, you know, for a person? For example, tall, short, so on and so forth.
Right, okay, so let's...
One more question.
Yeah. Let me explain something fundamental. Uniqueness in forensic genetics doesn't come from useful parts of DNA. Why? Because useful parts of DNA are very similar within the population. Uniqueness comes from useless pieces of DNA, which is known also as repetitive DNA. So what you have, your question, is going not towards a biological identification, but more or less in what we call phenotyping. And provided, for example, that we have, let's say, the Scandi working, single cells, with another process known as whole genome amplification, we should be able to get the whole genome information from that particular cell.
Because this links to what we're also doing, but in answer to your question, a lot of what is done traditionally is termed forensic genetics. What you're asking about is called forensic genomics. What can we do with the genome level data? You can start using, you know, SNPs to identify eye color, hair color. You can start to use these other phenotypic traits to ask more probative questions about the material you're collecting. And this is something that's already done. There are validated panels of markers used on what are called next generation or massively parallel instrumentation that answers those questions. It's just quite expensive, and it's not always needed in every case. Sometimes you need the old stuff, the stuff that just tells you it matches.
Yeah, it doesn't help if you say that the person had blue eyes, because in the population might be like this. Might be 5 people with blue eyes. So it's for forensics. That doesn't help
But what
much.
it's also going to help is not only tell us who it was, but it will tell us about the bacteria, the fungi, the archaea, the environment that person had come from. And that's very useful intelligence.
As soon as you start doing metabarcoding as
Absolutely.
well, you
That's
start getting a
metabarcoding.
phenomenal amount of information.
Yeah, this is going on too.
Oh, we can sort it out. Right, next question. Let's see who we got. Oh, it's right at the back. Right. Right, we'll do a chain. Dante, to you first. Here we go.
So if you have your Scandi thing working, and you can identify just from one cell the DNA of somebody, if I've taken just a wee tiny bit of blood from everybody here, and we've got it in a nice wee mixed sample, and then I've gone off and murdered somebody, left a drop of blood, are you going to have to test every cell that's in that sample? Because it could be any of us that's killed
I think one of the favourite pastimes of forensic scientists is to try and figure out how you can get away with stuff. Right? And what you're talking about there, you know, is the kind of late night, 2 o'clock in the morning conference, pub conversations that we have. The more sensitive tests become, the more difficult it is to separate, you know, truthful ground truth data from anything else, which is what you're talking about. You know, as soon as we are required to dismiss everybody else before we get to identifying the one person of interest, it's going to massively complicate it. So there's a lot of work to be done with the Scandi project to start answering some of those questions. But yeah, it's a very good point. I don't have an answer for you at the minute.
Yeah,
And it's not just who it is, but how did that trace get there?
Yeah.
We're talking about working at the source level information, but what really is needed by DI Jenkins is the activity level. So how did it get there?
Thank you. Next question. We've got time for a couple more. Oh, right in the middle.
So I was wondering about twins. So if it's identical twins, can you tell them apart with the samples or not?
The common, mostly we say that they have exactly the same DNA. I will give you some niche information. There are certain small differences. It's a very, very specialized research which actually has to do with the Y chromosome. And they have done these types of studies, but the differences are very, 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 very small. And with this type of analysis, you won't find different profiles.
Yeah, if you're using the traditional analysis, you wouldn't find it.
You need to
have
do
go to
something
that
very...
genome level information that we were talking about. You actually have to start looking more expansively. But there are genetic differences between identical twins, completely. You know, and you know, there has been famous cases where you've been able to use some of those more advanced techniques to differentiate between these identical twins. So it is possible. It's just difficult and expensive.
What we have to remember as well is our DNA has changed over time with our environment. What we eat, where we go, what we do. So there will be differences due to these things between two twins.
Yeah.
So not our normal techniques, but there are little tiny markers.
It's what we call epigenetic techniques.
Yeah. Did not know that at all. Right, fantastic. I can go for this. Here, ready? Very good.
How do you quantify uncertainty and communicate that? Because science isn't about certainty.
Absolutely, it's not categoric, none of it. So we have to deal with uncertainty, and how we deal with that is by using probabilities.
Yeah.
So we use Bayesian. Are there any statisticians in tonight? But we use that and we express that uncertainty. So
likelihood ratios, basically.
Yes. So just because you've got two profiles that match, you know, you're then interested in the statistical likelihood of that match. So that's when we actually start having to pose those statistical matches in a probability statement. The chance of another individual randomly selected from the population that matches that profile is 1 in a billion. So it's captured in the way we phrase the statements and the way that we run the statistics.
It's very similar to entomology, although we don't use statistics. That's why when I was speaking earlier, I said it was at least 10 days. We don't say it was at 2 p.m. on Sunday the 14th. We will say it's at least a time frame. We don't even give windows. It's always a minimum. We can calculate fairly accurately how long those insects have taken to develop to the stage that we collect them. But where that body has been beforehand, how long those insects have taken to find it, we can only predict. So we will always say, with a reasonable degree of confidence, it's a minimum of this. Body could have been in the freezer, it could have been in a suitcase, a wheelie bin. People like putting them in wheelie bins, I don't know why. Really stinky.
And suitcases.
And suitcases, but flies can lay their eggs through suitcases. We've watched them do it, we're that sad.
Wow.
There are videos of flies laying through suitcases, it's incredible.
But you've made a very good point about the communication of it. So that some of these issues, when you're all jurors, you potential jurors, so we have to think how we express that in the court.
That's probably the most difficult part to actually communicate with a juror. Basically because to actually explain to a common folk what Bayesian theorem is, I mean, it's difficult to explain to our students, and sometimes it's difficult to explain it to ourselves so we can deliver it to our students, and the phrasing you actually need to use, because that's also complex, and that's where Scandi comes a little bit useful.
Well, thankfully, there are also courtroom primers as well. So these are kind of primer documents that really break it down in quite a relatively easily digestible way that, you know, can get given to the legal system to kind of help, you know, bring down what we're saying to a more manageable and understandable level as well.
Yeah.
And this science communication is why organisations like Edinburgh Science exist. Thank you. I love that for a segue. Unfortunately, that is all we've got time for. The team here have been amazing. This morning they were running workshops for...
One more question.
You can come chat to the team at the end. We do need to everybody get going, so sorry. We literally are out of time. So this team have been amazing. They were doing workshops with kids this morning. They've done an amazing job. Can we give them a massive round of applause? Thank you so much.
Yeah, well, thank you again for such a fascinating fascinating talk. I was gripped by all of it, and brilliant acting there by D.I. Jenkins there. Thank you so much for, again, spending your Friday evening with us here at the Edinburgh Science Festival. We'll let you go into the night. Thank you so much for spending your evening. And one last time, big round of applause for our panel.